In this section, we will talk about interferons in detail. As we have discussed that, fortunately, there are other innate system weapons which are useful against virus-infected cells. Indeed, the innate system weapon that viruses fear most is the interferon system. This defense is so potent that most viruses have evolved ways to try to fend off the interferon system, at least long enough for the virus to reproduce and spread to a new host. Now, what are interferons? Well, interferons are proteins made and released by host cells in response to the presence of pathogens such as virus, bacteria, parasites, and tumor cells. They allow communication between cells to trigger the protective defenses of the immune system that eradicate pathogens and tumors. Interferon belongs to the large class of glycoproteins known as cytokines that are important in cell signaling. They are released by cell and affect the behavior of other cells, and sometimes the releasing cell itself. The virus, when affects the cell, it then multiple in cell. On a virus attack, cells can produce warning proteins called interferon alpha and interferon beta. Interferons are named so because these are proteins that can interfere with viral replication. Interferon alpha and interferon beta are called type 1 interferons, to distinguish them from interferon gamma, which is a type 2 interferon, which will be discussed later. Most human cells quickly produce type 1 interferons when they are attacked by a virus and have receptors on their surface for these interferon proteins. Consequently, the interferon produced by these cells can actually bind to receptors on the infected cell itself. This binding can result in the production of several hundred antiviral proteins that act to reduce the amount of virus produced by the infection. Even better, when the type 1 interferons produced by virus-infected cells bind to interferon receptors on nearby cells. Those cells are warned that there are viruses in the area and that they may soon be attacked. As a result of this early warning, the alerted cells turn on expression of antiviral genes and prepare to commit suicide if the virus does infect them. The elegant part of the interferon warning system is that, although the binding of interferon to its receptors prepares an uninfected cell for a viral attack, that cell continues to do business as usual unless an attack actually occurs. The alerted cell will only commit suicide if it is infected by a virus. Of course, for the cell, this is an altruistic act, because both the cell and the virus die together. Nevertheless, this beneficial suicide does prevent the virus from reproducing and infecting other cells. And if the attack does not come, the warned cell eventually stands down from its state of readiness. Although many cell types can produce type 1 interferons, the king of interferon by far is a white blood cell called a plasmacytoid dendritic cell. Plasmacytoid dendritic cell dedicate more than half of their protein-making capacity to making interferon. As a result, a plasmacytoid dendritic cell can make up to 1,000 times as much type 1 interferon per day as any other cell type. Consequently, these interferon factories can play a critical role in the innate system's defense against viruses, especially early in a viral infection. Let's discuss the toll-like receptor to understand the expression of interferon. There are 10 expressed toll-like receptor genes in humans and 12 in mice. Each toll-like receptor is devoted to recognizing a distinct set of molecular patterns that are essentially not found in healthy vertebrate cells. Initially called pathogen-associated molecular patterns, or PAMPs. These molecules are general components of both pathogenic and non-pathogenic microorganisms, and so are sometimes called microbial-associated molecular patterns. For example, galactose from self-antigen or beta-glucan from bacteria. In many viral infections, a double-stranded RNA intermediate is part of the viral life cycle, and frequently the viral RNA contains modifications that can be used to distinguish it from normal host RNA species. There are different types of toll-like receptors. For example, toll-like receptor 1, toll-like receptor 2, and so on. But in this video, we will only talk about toll-like receptors, which are involved in providing protection against viruses. Toll-like receptor 3 is expressed by macrophages, conventional dendritic cells, and intestinal epithelial cells. Toll-like receptor 7, toll-like receptor 8, and toll-like receptor 9, like toll-like receptor 3, are endosomal nucleotide sensors and are also involved in the recognition of viruses. Toll-like receptor 7 is expressed by plasmacytoid dendritic cells, B cell, and eosinophil. And toll-like receptor 9 are also expressed by plasmacytoid dendritic cells, B cells, and eosinophils. Toll-like receptor 8 is expressed primarily by monocytes and macrophages. Toll-like receptor 7 and toll-like receptor 8 are activated by single-stranded RNA. Toll-like receptor 3 is recognized by double-stranded RNA. Toll-like receptor 9 recognizes unmethylated CpG denucleotides. In mammalian genomes, CpG denucleotides in genomic DNA are heavily methylated on the cytosine by DNA methyltransferases. But in the genomes of bacteria and many viruses, CpG denucleotides remain unmethylated and represent another pathogen-associated molecular pattern. CpG is shorthand for cytosine and guanine separated by only one phosphate group. Phosphate links any two nucleosides together in DNA. Now, let's understand expression of antiviral interferons in response to viral nucleic acids can be stimulated by two different pathways from different toll-like receptor. Let's discuss the pathways. Signaling by mammalian toll-like receptors in various cell types induces a diverse range of intracellular responses that together result in the production of inflammatory cytokines, chemotactic factors, antimicrobial peptides, and the antiviral cytokines such as interferon alpha and interferon beta, which are the type 1 interferons. The nucleic acid sensing toll-like receptor, such as toll-like receptor 3, toll-like receptor 7, toll-like receptor 8, and toll-like receptor 9, activate members of the interferon regulatory factors family. Interferon regulatory factors proteins reside in the cytoplasm and are inactive until they become phosphorylated on serine and threonine residues in their carboxy termini. They then move to the nucleus as active transcription factors. Of the 9 interferon regulatory factors family members, interferon regulatory factor 3 and 7 are particularly important for toll-like receptor signaling and expression of antiviral type 1 interferons. For toll-like receptor 3, expressed by macrophages and conventional dendritic cells, and the toll-like receptor 3 recognize the double-strand RNA of virus. So cytoplasmic toll-interleukin-1 receptor domain of toll-like receptor 3 interacts with the adapter protein. Toll-interleukin-1 receptor domain containing adapter-inducing interferon beta. Toll-interleukin-1 receptor domain containing adapter-inducing interferon beta interacts with the E3 ubiquitin ligase tumor necrosis factor receptor associated factor 3. which, like tumor necrosis factor receptor-associated factor 6, generates a polyubiquitin scaffold. In toll-like receptor 3 signaling, this scaffold recruits a multi-protein complex containing the kinases IKK epsilon and tank-binding kinase 1, which phosphorylate interferon regulatory factor 3. Toll-like receptor 4 also triggers this pathway by binding toll interleukin 1 receptor domain containing adaptor-inducing interferon beta. But the interferon regulatory factor 3 response, induced by toll-like receptor 4, is relatively weak compared with that induced by toll-like receptor 3, and its functional role in vivo remains elusive. It then moves into the nucleus and gives rise to the expression of type 1 interferon genes. In contrast to toll-like receptor 3, toll-like receptor 7, toll-like receptor 8, and toll-like receptor 9 signal uniquely through myeloid differentiation factor 88. For toll-like receptor 7, signaling in plasmacytoid dendritic cells, and it recognizes the single standard RNA. The myeloid differentiation factor 88 toll interleukin 1 receptor domain recruits the interleukin 1 receptor-associated kinase 1 and interleukin 1 receptor-associated kinase 4. Here, the interleukin 1 receptor-associated kinase complex carries out a distinct function beyond recruiting tumor necrosis factor receptor-associated factors that generate a signaling scaffold. In these cells, interleukin 1 receptor-associated kinase 1 can also physically associate with interferon regulatory factor 7, which is highly expressed by plasmacytoid dendritic cells. This allows interferon regulatory factor 7 to become phosphorylated by interleukin 1 receptor-associated kinase 1, leading to induction of type 1 interferons. Not all interferon regulatory factor regulate type 1 interferon genes. Interferon regulatory factor 5, for example, plays a role in the induction of pro-inflammatory cytokines. The collective ability of toll-like receptors to activate both interferon regulatory factors means that they can stimulate either antiviral or antibacterial responses as needed. In human interleukin 1 receptor-associated kinase 4 deficiency, for example, no extra susceptibility to viral infections has been noted. This would suggest that interferon regulatory factor activation is not impaired and the production of antiviral interferons is not affected. Toll-like receptors are expressed by different types of cells, involved in innate immunity, and by some stromal and epithelial cells. And the responses generated will differ in some respects, depending on what type of cell is being activated. There are other pathways for expression of type 1 interferon genes, such as cytosolic DNA sensors signal through stimulator of interferon genes, or STING, to induce production of type 1 interferons. Let's understand this pathway to understand the induction of type 1 interferons. This is cell. Virus enters the cell. Double-stranded DNA from viruses activates cyclic guanosine monophosphate adenosine monophosphate synthase to produce cyclic guanosine adenosine monophosphate from ATP and GTP. Innate sensors that recognize cytoplasmic RNA use specific modifications, such as the 5-cap, to discriminate between host and viral origin. Host DNA is generally restricted to the nucleus, but viral, microbial, or protozoan DNA may become located in the cytoplasm during various stages of infection. CGAMP, or cyclic guanosine adenosine monophosphate, bind to the STING dimer present on the endoplasmic membrane and activate its signaling. Several innate sensors of cytoplasmic DNA have been identified that can lead to the production of type 1 interferon in response to infections. STING activates the kinase tank-binding kinase 1 to phosphorylate interferon regulatory factor 3, which enters the nucleus and induces expression of type 1 interferon genes. One component of the DNA sensing pathway is STING, or stimulator of interferon genes. It was identified in a functional screen for proteins that can induce expression of type 1 interferons. STING is anchored to the endoplasmic reticulum membrane by an amino-terminal tetraspan transmembrane domain. Its carboxy-terminal domain extends into the cytoplasm and interacts to form an active STING homodimer. Now, many cytokine receptors signal using a rapid pathway called the JAK-STAT pathway. Let's discuss JAK-STAT pathway. This is cell. This is nucleus of cell. While these are cytokines. Cytokine receptors consist of at least 2 chains, the cytoplasmic domains of which bind Janus kinases receptor consists of at least 2 chains, each associated with a specific JAK. Next step is that cytokine binding dimerizes the receptor, bringing together the cytoplasmic JAKs, which activate each other and phosphorylate the receptor, then to phosphorylate specific tyrosine in the receptor tails. The STAT, or signal transducer, and activator of transcription family of proteins have an N-terminal domain that homodimerizes STATs in the cytosol before activation, and an SH2 domain that binds to the tyrosine phosphorylated receptor tails. This is an active STAT dimer. In next step, transcription factors that are STATs homodimers bind to the phosphorylated receptors and are in turn phosphorylated by the activated JAKs. After phosphorylation, STAT proteins reconfigure into a dimer that is stabilized by SH2 domain binding to phosphotyrosine residues on the other STAT. It is active STAT dimer. Now, they then translocate to the nucleus, where they bind to and activate the transcription of a variety of genes important for adaptive immunity. There are 7 STATs, which are 1 to 4, 5A, 5B, and 6, which reside in the cytoplasm in an inactive form until activated by cytokine receptors. Before activation, most STATs form homodimers due to a specific homotypic interaction between domains present at the amino termini of the individual STAT proteins. As we have discussed that how interferons are produced by different pathways. Now, we will have an overview of antiviral role of interferons. The interferons, which are interferon alpha and interferon beta, have 3 major functions. First, they induce resistance to viral replication in uninfected cells by activating genes that cause the destruction of messenger RNA and inhibit the translation of viral proteins and some host proteins. These include the MX proteins, oligoadenylate synthetase, and double-stranded RNA-dependent protein kinase. And secondly, they can induce MHC class I expression in most cell types in the body, thus enhancing their resistance to natural killer cells. They may also induce increased synthesis of MHC class I molecules in cells that are newly infected by virus, thus making them more susceptible to being killed by CD8 cytotoxic T cells. Third, they activate natural killer cells, which then selectively kill virus-infected cell. Let's understand the mechanism for inhibition of translation of viral proteins by MX proteins or myxoma-resistant proteins, oligoadenylate synthetase, and double-stranded RNA-dependent protein kinase. One interferon-stimulated genes encodes the enzyme oligoadenylate synthetase, which polymerizes ATP into 2,5-linked oligomers, whereas nucleotides in nucleic acids are normally linked 3,5. These 2,5-linked oligomers activate an endoribonuclease that then degrades viral RNA. A second protein induced by interferon alpha and interferon beta is a double-stranded RNA-dependent protein kinase called PKR. This serine threonine kinase phosphorylates the alpha subunit of eukaryotic initiation factor 2, thus suppressing protein translation and contributing to the inhibition of viral replication. MX, or myxomer-resistant proteins, are also induced by type 1 interferons. Humans and wild mice have two highly similar proteins, MX1 and MX2, which are GTPases, belonging to the dynamin protein family. But how they interfere with viral replication is not understood. Oddly, most common laboratory strains of mice have inactivated both MX genes, and in these mice, interferon beta cannot act to protect against influenza infection. This was all about the role of interferons in innate immunity against viruses. In our next session, we will talk about the role of natural killer cell against viruses in innate immunity.